Hi everyone, today we will be talking about MHC1 antigen processing and presentation. In the last class, we discussed about structure, function and expression of MHC1 and MHC2 molecules. Today we will focus on antigen processing and antigen presentation. We also focus on peptide loading onto MHC molecule. What is antigen processing? Antigen processing is a process in which larger molecules such as proteins are broken down into small pieces called peptides. These peptides, once they are formed, they are presented by MHC molecule and displayed on antigen presenting cells. And this process is called antigen presentation, so that antigens are recognized by T cells. This slide shows the processing of peptide antigens for MHC class 1 presentation. So cytosolic proteins are intracellular antigens. It could be either viral protein or cell proteins. They are first folded, unfolded, and ubiquitinated and degraded in proteasomes. So this slide shows the detailed pathway of class 1 MHC pathway of processing of cytosolic antigens. So it is a multi-step process. Initially, there is a production of proteins in the cytosol. As I mentioned, initially is viral proteins, synthesized viral proteins, first ubiquitinated and degraded in proteasomes. After trimming by cytosolic proteases, these peptides enter the endoplasmic reticulum by a special molecule called TAP. TAP has two subunits, TAP1 and TAP2, and this is the central core part of peptide loading complex. So as you can see, this is the MHC class 1 MHC alpha chain. It is a linear peptide in the ER. Initially formed as a linear peptide in the endoplasmic reticulum, now it is folded with the help of many chaperone molecules, including calnexin, calreticulin, BIP, that is binding immunoglobulin protein, and endoplasmic reticulum protein 57, ERP 57. During this process, beta-2M chain is also added to the alpha chain. Now the process is complete. MHC1 molecule is formed. This complex is held together by another molecule called tapasin, which facilitates the transfer of peptides to the antigen binding cleft. And so this everything happening in endoplasmic reticulum. Another second chaperone called TAP BPR can further optimize the peptides in MHC. And now what happens? This peptide loaded. Now MHC has a peptide loaded already here. This is transferred to Golgi and then transported to surface so that T cell can recognize it. So as I mentioned, it has 4 different steps. Initially, production of proteins in the cytosol and proteolytic degradation of proteins, and those peptides are transported from cytosol to endoplasmic reticulum, and then assembly peptide 1, peptide and class 1 MHC form a complex in endoplasmic reticulum, and once the complex is formed, it is also transferred to Golgi and further transferred to on the membrane, plasma membrane, so that the T cell can recognize it. So this is again the different steps included in the process. We already discussed this part. So this cartoon shows the different steps included in the process, how to present your inner self. So as you can see, large molecular proteins are degraded by proteasome. Now peptides are formed. These peptides are transported by TAP and now tapasin facilitates the transfer. And now another chaperone called a TAP BPR, it trims the peptides. And now only the correctly trimmed and correctly formed peptides are transported to MHC. And now this here you can see MHC one peptide complex is recognized by cytotoxic T lymphocytes. So finally, we have questions here. Do you have any questions? And this is the question that you can answer. It's a simple question. Proteasomes play a key role in processing. Answer is endogenous antigens. Okay.